Hello viewers, in today's session under Food Technology, we will be learning about water soluble vitamins with special reference to their structure, function and stability. Vitamins are a diverse group of organic compounds that are nutritionally essential micronutrients. They are present in small amounts in food. They are susceptible to change in structure and stability when the environmental conditions are not favorable. Losses of vitamins occur during processing, handling, packaging, exposure to heat, pH, presence or absence of oxygen and cooking. In the human system, they act as coenzymes and precursors in the metabolism of macronutrients. Vitamin C helps in reducing the oxidative stress caused by free radicals by neutralizing their action. Vitamins are essential for maintenance of overall health. When the body is deficit in one or more vitamins, they lead to diseases. To know more about the importance of water soluble vitamins, the session is divided into 5 major subdivisions, namely water soluble vitamins, functions and their form, structure of water soluble vitamins, stability of water soluble vitamins, bioavailability of water soluble vitamins and other vitamin like compounds. Firstly, water soluble vitamins, functions and their forms. Water soluble vitamins are the member of B complex vitamins along with vitamin C. They are ascorbic acid, thiamine or B1, riboflavin or B2, niacin or B3, pantothenic acid or B5, pyridoxine or B6, folic acid or B9, biotin and cobalamin, which is B12. The sources of vitamins in general are from plants, animals, microorganisms and fortified food products. The common feature of these vitamins is their solubility in water. Most of the water soluble vitamins cannot be stored in the body and hence their regular intake is of utmost importance. Thiamine. Different forms of thiamine are free thiamine, thiamine monophosphate and thiamine pyrophosphate or thiamine diphosphate, thiamine triphosphate, adenosine thiamine triphosphate and adenosine thiamine diphosphate. Thiamine pyrophosphate is the active form. Their activation requires phosphorylation by kinases into the diphosphate and triphosphate esters. Thiamine pyrophosphate is essential for conversion of pyruvate to acetyl-CoA to generate energy. It is a coenzyme for transketolase in the oxidation of glucose by the hexose monophosphate shunt. Most of the circulating thiamine is bound to proteins such as albumin and erythrocytes. Their absorption is by active transport or passive diffusion. Riboflavin. It is essential in tissue respiration, growth and is a transporter of hydrogen ions. Different forms of riboflavin in the system are free riboflavin, flavin adenine dinucleotide or FAD and flavin adenine mononucleotide or FMN. The coenzyme forms of riboflavin, that is FAD and FMN, are the active forms which involve the action of flavokinases. These forms are essential in the initial oxidation of fatty acids and several intermediates of glucose metabolism. Their activation requires hydrolytic cleavage of free riboflavin from its flavoprotein complexes by various phosphatases. As thiamine, riboflavin is also bound to plasma proteins such as albumin. Its absorption is by a carrier mediated process which requires adenosine triphosphate or ATP. Niacin. It is essential in glycolysis, fat synthesis and tissue respiration. The different forms of niacin are nicotinic acid and nicotinamide. It is very essential for energy production and metabolism of cells. It is part of the pyridine nucleotide coenzymes nicotinamide adenine dinucleotide or NADH or nicotinamide adenine dinucleotide phosphate, which is NADPH, which facilitate transport of hydrogen. These molecules are co-substrates for more than 200 enzymes involved in metabolism of carbohydrates, amino acids and fatty acids. It can be synthesized from tryptophan. Most of the food have NADH and NADPH, which undergo digestion to release the absorbable forms nicotinamide and nicotinic acid. These forms are absorbed in the intestine by carrier-mediated facilitated diffusion. Niacin in the plasma is not bound to any molecule and is taken up by tissues through passive diffusion. Few tissues have transport system for nicotinic acid. In the tissue, it is reconverted to NADH and NADPH. Pantothenic acid. It is essential in the intermediary metabolism of carbohydrates, fat and protein. It is an integral part of coenzyme A or CoA, which is involved in energy production from the macronutrients and acyl carrier protein, or ACP, which is used in synthesis reaction. CoA is important in energy metabolism for pyruvate to enter the tricarboxylic acid cycle or the TCA cycle as acetyl-CoA, and for alpha ketoglutarate to be transformed to succinyl-CoA in the cycle. The CoA and ACP forms are found in foods and their absorption requires hydrolysis to phosphopantothene and later the conversion to pantothenic acid. It is absorbed by passive diffusion and active transport in the jejunum. It is transported in its free acidic form in the plasma and taken up by diffusion into erythrocytes. Later, the peripheral tissue take up pantothenic acid by sodium dependent active transport and in few tissues by facilitated diffusion. Pyridoxine. It comprises of three forms: the alcohol derivative pyridoxine, the aldehyde derivative pyridoxal, and the amine pyridoxamine. Pyridoxal phosphate is the active form. It is formed from pyridoxal by phosphorylation catalyzed by pyridoxal kinase. However, the absorption into the jejunum and ileum is in their dephosphorylated form. Pyridoxal phosphate are bound to protein and act as prosthetic group or coenzyme of enzymes which are involved in transamination, decarboxylation, transsulfuration, desulfuration and non-oxidative deamination reactions, primarily in metabolism of proteins. Pyridoxal phosphate is coenzyme for enzymes that are involved in the synthesis of heme, neurotransmitters, catecholamines and coenzyme A synthesis. It is essential in the biosynthesis of niacin from tryptophan and unsaturated fatty acids from essential fatty acids. Folates. Dietary folates are absorbed as monoglutamate methylated forms, which are 5-methyl tetrahydrofolic acid and 5,4-myl tetrahydrofolic acid. Absorption occurs at the jejunum by active transport or by passive diffusion when ingested in large amounts. Dietary folates exist as polyglutamyl form with several glutamate residues, whereas folic acid, the synthetic form, is an oxidized monoglutamate form. Polyglutamates are hydrolyzed by conjugases to corresponding monoglutamates for intestinal absorption. Once they are absorbed, they are converted into 5-methyl tetrahydrofolate, which is the major circulating form of folates in the body. The different forms of tetrahydrofolic acid act as enzyme co-substrates in synthesis reactions in the metabolism of amino acids such as methionine, formation and maturation of red and white blood cells, and for biosynthesis of RNA and DNA as single carbon atom donors or acceptors. It is vital during early fetal development. Vitamin B12. It functions in two metabolically active forms, adenosylcobalamin and methylcobalamin. However, other forms are cyanocobalamin and hydroxycobalamin. They play a major role in the metabolism of propionate, amino acids and single carbons. They are synthesized by the microflora of the gut, but it cannot be absorbed. A deficiency causes impaired cell division through arrested synthesis of DNA. Therefore, they are essential for the biosynthesis of nucleic acids and nucleoproteins. In the food system, it is usually bound to proteins and is released by the action of pepsin. It combines with cobalamin in the stomach and moves to the small intestine. The cobalamin detach from cobalamin by hydrolysis. Cobalamin later gets bound to intrinsic factor, which is crucial since majority of B12 is absorbed by this active transport. Biotin. Biotin in foods is largely bound to protein and is released by proteolytic digestion to yield free biotin, biocytin or biotinipeptides. By the action of biotinidase, biotin is released from biocytin and biotinipeptide. Free biotin is absorbed in the proximal small intestine primarily by carrier-mediated diffusion and transported into the plasma. Biotin is mainly a carboxyl carrier and acts by addition or removal of carbon dioxide and is involved in the gluconeogenesis and fatty acid synthesis. It removes the amine group from amino acids. Ascorbic acid. Ascorbic acid or vitamin C functions in oxidation reduction reaction. Humans receive ascorbic acid from their diet, which is absorbed by active transport or passive diffusion. Two forms of vitamin C exist in the system: dehydroascorbic acid, which is the oxidized form, and the reduced form ascorbate or ascorbic acid. The former is better absorbed. It is involved in the synthesis and production of collagen, neurotransmitters, serotonin and norepinephrine, interferons conferring immunological potential, and carnitine. It is an efficient antioxidant and prevents oxidative damage. It converts ferric to ferrous state of iron, thus facilitating iron absorption overall and from the non-heme sources. It is also important in wound healing. The next topic of discussion is the structure of water-soluble vitamins. Thiamine. It is named as a thiovitamin or sulfur-containing vitamin. It is a colorless organosulfur compound with the chemical formula C12H17N4OS. Its structure consists of an aminopyrimidine and a thiazole ring linked by a methylene bridge. The thiazole is substituted with methyl or hydroxyethyl side chain as displayed on the screen. Riboflavin. The name is derived from ribose, which is a sugar, and flavus, which means yellow. The ring moiety flavin imparts yellow color. Riboflavin appears as yellow-orange crystals. The molecular formula is C17H20N4O6. Riboflavin has 3 benzene rings and 6 tetrahedron bond shapes as displayed on the screen. Niacin. It is a colorless compound which is a derivative of pyridine with a carboxylic acid at C3 position. Nicotinamide has a carboxamide or CONH2 group instead of carboxylic acid. The molecular formula of niacin is C6H5NO2. Pantothenic acid. It is the amide between pantoic acid and beta-alanine. The molecular formula of pantothenic acid is C9H17NO5. Pyridoxine. By replacing the hydroxymethyl group on position 4 of the pyridoxine molecule with an amine and aldehyde respectively, two related compounds, pyridoxamine and pyridoxal, can be formed, which are also vitamin active. The molecular formula of pyridoxine is C8H11NO3. Biotin. Biotin in nature appears as white crystalline needles. It is composed of an urido or tetrahydroimidazolone ring fused to a tetrahydrothiophene ring with a valeric acid side chain. Tetrahydrothiophene is a 5-membered ring containing of 4 carbon atoms and 1 sulfur atom. The molecular formula is C10H16N2O3S. Vitamin B12. The molecular formula of vitamin B12 is C63H88CO N14O14P. The structure of B12 is based on Corin ring. The ring consists of 4 pyrrole subunits joined on opposite sides by a CCH3 methylene link on one side by a CH methylene link and with the 2 of the pyrroles joined directly. It is thus like a porphyrin but with one of the bridging methylene groups removed. The nitrogen of each pyrrole is coordinated to the central cobalt atom. The central metal ion is cobalt. 4 of the 6 coordination sites are provided by the Corin ring and the 5th by a dimethylbenzimidazole group. The 6th coordination site, the center of reactivity, is variable, being a cyano group, a hydroxyl group, a methyl group or a 5-deoxyadenosine group. Here the carbon 5 atom of the deoxyribose forms the covalent bond with cobalt respectively to yield the 4 B12 forms: cyanocobalamin, hydroxycobalamin, methylcobalamin and adenosylcobalamin respectively. The structure of B12 is the most complex of all vitamins. Folate. The molecular formula of folate is C19H19N7O6. Folic acid consists of a pyridine nucleus, p-aminobenzoic acid and glutamate. In their reduced form, cellular folates function as conjugated to a polyglutamate chain. These folates are a mixture of unsubstituted polyglutamyl tetrahydrofolates and various substituted 1-carbon forms of tetrahydrofolate, for example 10-formyl, 5,10-methylene and 5-methyl form. The reduced forms of the vitamin, particularly the unsubstituted dihydro and tetrahydro forms, are unstable chemically. They are easily split between the C9 and N10 bond to yield a substituted pyridine and a p-aminobenzoyl glutamate, which have no biological activity. Vitamin C. L-ascorbic acid is a carbohydrate-like compound whose acidic and reducing property are contributed by the 2,3-enediol moiety. It is highly polar and readily soluble in aqueous solution. Ascorbic acid contains 2 optically active isomeric forms, C4 optical isomer D-ascorbic acid and C5 optical isomer L-isoascorbic acid, which is also known as erythorbic acid. The molecular formula is C6H8O6. Stability of water soluble vitamins. Physical methods. Processing such as milling and refining has shown decrease in water soluble vitamins such as pantothenic acid, riboflavin, biotin, niacin, thiamine, folic acid and vitamin B6, which are concentrated in the germ and bran of the grain. Polishing of grains significantly reduces B vitamins. They are easily washed out during food storage and processing. Refrigeration can minimize the loss of water soluble vitamins. Preliminary treatments such as trimming and washing of fruits and vegetables causes loss of vitamins present in the peel, skin or the discarded unedible part of the food. Cuts, damage, washing and cooking will leach water soluble vitamins. Temperature. Thermal processes such as blanching, which is an essential step during processing of fruits and vegetables to inactivate enzymes and to reduce microbial load on the surface, has shown larger losses of water soluble vitamins. Canning, which involves blanching as the preliminary step, has shown loss of folate, pantothenic acid, thiamine, vitamin C, riboflavin and niacin. Thiamine is heat sensitive in neutral and alkaline foods and is unstable in air. High temperature short time retains vitamin than low temperature long time. Microwave heating has the potential for a greater retention of heat labile vitamins compared with other more conventional methods because the heating time is shorter. Retention of vitamin C, vitamin B1, B2 and B6 is higher in microwave heating than in conventional heating methods. Freezing does not cause loss of vitamins. Moisture. Water activity greatly influences the stability of vitamins. Higher the water activity, greater is the degradation rate. The process, temperature and exposure time decides the extent of loss of water-soluble vitamins in dehydrated foods. Direct contact of the food material to the heated surface causes higher loss of water-soluble vitamins than indirect methods such as spray drying. Oxygen. Vitamins sensitive to oxidation such as ascorbic acid, folate and thiamine are lost during drying of foods. Chemicals. Chemicals such as chlorine, sulfites, nitrites interact with vitamins by oxidation and reduction to inactivate water-soluble vitamins. Sulfite ions directly react with thiamine causing inactivation. Sulfites also react with carbonyl groups and convert active vitamin B6 aldehydes to their inactive sulfonated derivatives. Ascorbic acid prevents formation of N-nitrosamine in nitrite treated meat. The stability of ascorbic acid and thiamine is higher at lower pH, and higher pH decreases the stability of ascorbic acid, thiamine, pantothenic acid and folates. Light. Vitamin C, riboflavin, pyridoxine, folic acid and vitamin B12 become unstable when exposed to light. Irradiation. Irradiation causes loss of thiamine, whereas riboflavin and vitamin C shows better stability. The overview of stability of water-soluble vitamins in different media is given in Table 1 as displayed on your screen. Bioavailability of water-soluble vitamins. Bioavailability refers to the degree to which an ingested nutrient undergoes intestinal absorption and metabolic function or utilization within the body. Bioavailability depends on the concentration of the vitamin present during consumption, composition of the diet, intestinal transit time, efficiency of the intestine to absorb nutrients, enzyme concentration, rate of conversion of vitamins to its active form and interaction of vitamins with other food components such as proteins, carbohydrates and fat. Various studies have been conducted to improve the absorption of vitamins. In a study conducted by Vinson et al., a significant improvement was noted in the plasma levels of vitamin E and C when these vitamins were consumed with aloe vera preparations. Fortified foods and supplements are effective than high folate foods when it comes to bioavailability of folates. Therefore, added folic acid is better absorbed than natural folic acid. Diet high in fruits and vegetables possess better bioavailability of folates. Few anti-thiamine factors such as thiaminase 1 and 2 and polyphenols make thiamine unavailable for absorption. Dietary fiber improves the absorption of riboflavin. Chronic alcoholism decreases the bioavailability of most of the B vitamins. Other vitamin-like compounds. Compounds such as choline, carnitine, myoinositol, ubiquinone and bioflavonoids are classified under vitamin-like compounds. As the name suggests, they have characteristics of vitamins and are essential. Few of them such as carnitine can be biosynthesized and few such as choline have to be provided in the diet. Choline is a structural compound in the membrane phospholipids as phosphatidylcholine and in the neurotransmitter acetylcholine. Carnitine helps in energy production by the transport of long chain fatty acids into the mitochondria for oxidation in a process called carnitine transport shuttle. Myoinositol in the form of phosphatidylinositol serves as an anchor for membrane proteins by covalent bonding. It is essential for the biosynthesis of eicosanoids. They are important sources of intracellular signals and act as secondary cell messengers in response to hormonal stimuli. Ubiquinones are essential for the reversible redox reactions in the mitochondrial electron transport system. Bioflavonoids help in reducing the risk of cardiovascular diseases and cancers. To conclude, vitamins are essential components of the diet. Stability of vitamins depend on various factors such as temperature, processing method, light and radiation. The bioavailability of vitamins depend on the concentration of vitamin present during consumption, composition of the diet, intestinal transit time, efficiency of the intestine to absorb nutrients, enzyme concentration, rate of conversion of vitamins to its active form, and interaction of vitamins with other food components. Bioavailability can be affected by alcoholism or a non-functional gut due to the presence of a disease. Like vitamins, there are other compounds which are conditionally essential for the body. Thank you.